Okay, it is 9 o'clock, so I'll just welcome everyone. Thank you for joining the University of Washington Division of Rheumatology Grand Rounds. A couple of reminders before we start the session. This session is currently being recorded and it will be posted on our website later. There will be a Q&A session at the end, so please add your questions in the Q&A feature and not in the chat box so that it's easier to moderate. And now I will let Dr. Lude introduce our speaker for today.
Thank you, Danielle. And my apologies for my voice today. It's my great pleasure to introduce today's Grand Round speaker, Dr. Steffen Massberg, Professor and Chair of Cardiology at the University Hospital Munich, Germany. Professor Massberg received all of his medical training at the Ludwig Maximilians University in Munich and was board certified cardiologist in 2007, shortly after finishing up a 2-year research position at Harvard Medical School. Dr. Massberg has been highly prolific, publishing hundreds of seminal papers with a very impressive H index of more than 100, meaning that more than 100 of his publications have been cited more than 100 times. His research spans a broad area of cardiology, including clinical trials, platelet biology, mechanisms of thrombosis, and not least the crosstalk between immune cells and coagulation, with his studies being published in top-tier journals such as New England Journal of Medicine, Circulation, Blood, and Immunity. And today, Dr. Massberg will share with us some of his insights into the crosstalk between platelets and immune cells, which should be very interesting and highly relevant to many of the diseases we study as well. So with that said, Dr. Massberg, please take it away.
Thank you, Christian, for the kind invitation to speak here and also for the kind introduction. And welcome to everybody listening. So I can see only Christian, but I hope there's more than Christian. I will be talking about the collaboration between neutrophils with platelets and the role of this collaboration in thrombosis and beyond. As Christian mentioned, I'm a cardiologist and therefore one of the group of patients that I'm treating mostly is patients with acute myocardial infarction. You can see one example here on the left-hand side, a patient that is undergoing coronary catheterization for ST elevation myocardial infarction of the inferior wall. You see the right coronary artery is closed here at that section. And if you were to look at it by a microscope, you would see that this is actually a thrombus. We can look at it not in the human setting with the microscope, but in the mouse model. And this is one example here where we induced endothelial disruption, similar as it occurred here spontaneously in this patient. We did it with a wire injury or laser injury in this case, actually. An endothelial lesion here was induced, and this leads to a very rapid response with recruitment of initially individual platelets, and then these platelets recruit additional platelets, and within a couple of microseconds, a thrombus grows that can occlude this vessel here within a matter of a few seconds. However, thrombosis is not only triggered by platelets and fiber inflammation. Actually, if you stain for platelets, of course, you will only see platelets as shown here, but if you stain also for inflammatory cells, the same vessel will give you a completely different picture. So this is done here where we stain for neutrophils with the GO1 antibody, and you see actually within this thrombus, of course, you have a lot of platelets, but you also have a lot of neutrophils, and actually there's a very close physical contact between these two cellular compartments, the neutrophils in red and the green platelets. So this is the best basis for a very close and intimate collaboration. This is actually what our group and also, of course, many other groups across the world has shown in the last couple of years, that there is a strong collaboration between immunity and platelets as well as coagulation, triggering thrombus formation, and this jointly, this collaboration is termed thromboinflammation or immunothrombosis. This is not something that we see only in the mouse experimental setting, but we can actually see this collaboration also in the human setting. And one example is shown here, where we did multi-panel flow cytometry of human thrombi collected from cerebral arteries from stroke patients, and we analyzed the thrombi, and actually we found that in addition to platelets, which were the most abundant cell substance. We have also found a huge amount of inflammatory cells, with neutrophils being the most abundant population of immune cells within human thrombi, meaning that not only in the animal model system you have this close correlation, as I said before, but also in a human scenario. And actually, we will be able to use this analysis also in clinical scenarios, hopefully in future, to better characterize thrombi and also the origin of thrombi, which is interesting in the setting of stroke patients. So how is this collaboration working? Actually, several studies, particularly also from the lab of Denisa Wagner, but also from other work for studies all around the world, have shown that there's several mechanisms involved in the collaboration between platelets and neutrophils. One of the central aspect being the formation of nets, neutrophil extracellular traps. Platelets can support neutrophils in net formation, and we will come to this in a second, through several mechanisms. And these nets then are the very strong prothrombotic surface activating platelets, activating coagulation pathways, and thereby boosting thrombin formation. And this translates into fibrin formation, which is essential for propagation of thrombus formation in the arterial setting, but as we see also in the venous thrombus setting. So nets are a central element of thrombus formation in arterial thrombosis, and we have, together with other groups, looked at several of the mechanisms involved in this. Platelets can boost netosis, so they collaborate with neutrophils in netosis, and they do so by exposing several proteins on their surfaces, including HMGP1, platelet factor 4, and the chemokine CACL5. All of these can activate neutrophils to form nets, but there's also a positive feedback loop from activation of neutrophils linking to platelet activation with platelet pyroptosis that can also feed back again to neutrophil activation with netosis. So there's a very strong circuit of platelet activation leading to neutrophil activation, feeding back to platelet activation that culminates in the hyperactivation of neutrophils with the consequence of forming prothrombotic neutrophil nets that then are essential for thrombus propagation in the arterial thrombosis setting, as shown in the example of myocardial infarction. However, nets are not only relevant in arterial thrombosis, but also in the setting of venous thrombosis, which is probably also very interesting for the group of people that are listening today in the context of rheumatoid diseases, which is known to be linked to higher incidences of venous clot formation. Also there, we know that activation of nets is a key element that leads to thrombus propagation. However, one major difference in the setting of venous thrombosis compared to arterial thrombosis is that in the arterial setting, you have endothelial disruption as a major trigger, whereas in venous clot formation, reduced blood flow velocity, so reduction of blood flow, is a major trigger of venous, of the thrombotic cascade, culminating in netosis, leading then to thrombus formation. We know, and this is one thing that you probably also see a lot, that immobilization is a major trigger of venous thrombosis. So patients that are immobilized for a couple of days receive anticoagulants to prevent the risk of venous thrombosis. However, there are examples where even long immobilization does not lead to venous thrombosis. And this is something I wanted to share with you. One example are hibernating animals. The most prominent examples are bears. Bears undergo hibernation for several months, 6 to 7 months a year. And despite this hibernation, they are sleeping on this all the time for 6 months. Despite this very long mobilization, they do not develop mobilization. This is kind of appears to be a contradiction to the known increase in thrombotic risk in acutely immobilized patients. So how do bears and other hibernating animals avoid thromboembolism? And we looked at this more closely and analyzed one of the most prominent surrogate parameters of thrombus formation in hibernating bears. Bears compared wake bears during summertime to winter bears undergoing hibernation. And this is a collaboration with the Swedish bear project. So when we analyzed D-dimer formation in bears during wintertime, we saw there was a significant lower level of circulating D-dimers compared to summertime, providing a first evidence that indeed there appears to be happening something that protects hibernating immobilized bears from venous thromboembolism. So to look at this in greater detail, we, as I said, we collaborated with the Swedish bear or Scandinavian bear project. We thought this would be interesting because if we were able to come up with a mechanism that leads to a biological mechanism, actually, that leads to thromboprotection, this could be also, of course, from a therapeutic aspect, a very interesting novel target or even targets, depending on what we would find. So that's why we thought this would be an interesting model system to look at more closely. So what we did, in this collaboration, we compared active brown bears during summertime with the same bears during winter, during their hibernation. We took jugular venous blood samples, analyzed them locally, so on site, and also stored some samples. And from sampling to analysis, it took us 2 hours because, of course, the bears are not in this house, fortunately, but they are free bears running around or sleeping in their caves. So what we saw when we did a very superficial analysis of these bears, we did not see any relevant differences in correlation, also not in the expression of clotting factors. However, what we saw in the initial screen that during wintertime, platelet aggregation is suppressed. For example, here, low amounts of collagen stimulation. You don't see in blue here aggregation during winter, but you see a moderate aggregation during summertime. If you increase collagen concentration, you see a moderate response in winter, but a very strong response in summer. And the same is also true for low concentrations and higher concentrations of thrombin, indicating that platelets or the blood can be activated to aggregate, but it's less responsive during wintertime compared to summertime. And this is just showing the quantification. So a lower response to collagen regarding a maximum aggregation in winter. And the same is also true for in particular for low concentrations of thrombin and not so much for higher thrombin concentrations. So we wanted to see what is the mechanism behind it. And to cut a long story short, we analyzed several parameters, and it turned out that the major difference was platelet functionalities. So therefore, we looked at the platelet proteome more closely and compared active bears, blood platelets from active bears to blood platelets sampled from hibernating bears during wintertime. And what we found is actually that there is a number of proteins that are downregulated during wintertime, and there are some are also upregulated. And the most prominent protein that is regulated is HSP47. And actually, we found a 54-fold downregulation of this heat shock protein 47 in wintertime compared to summertime. And here, Western blots showing you 3 examples, 3 beers. So here one, no expression of HSP47. Second one, also no expression in platelets. And there's a very small, very moderate expression here in this third example of beer. So there appears to be a very reproducible downregulation, profound downregulation of heat shock protein 47 in platelets isolated from bears during wintertime compared to summertime. We wanted to see whether we can model a similar thing also in humans. And so to do that, we exposed 10 volunteers to a 27-day long immobilization with a 6-degree head-down positioning, and measured HSP47 expression compared to baseline. And as in long-term embolization in the hibernation phase of bears, we also saw a downregulation in these human healthy volunteers after 27 days. If we also sampled after 7 days, and there we did not see any downregulation. So it appears to take some time until downregulation shows up in the platelet compartment. So this shows that both in bears and in humans, HSP47 is strongly regulated, downregulated during immobility. And so this is a mechanism that appears to be conserved in different species. So of course, we wanted to see what the functional consequence of downregulation of HSP47 could be. And to do that, we used a mouse model where we knocked out HSP47 specifically in the platelet compartment of mice using PF4-Cre conditional HSP47 knockout mice. And if we did that and induced thrombosis, in this case arterial thrombosis or venous thrombosis, actually venous thrombosis in this case, sorry, we found a significant reduction of thrombus formation with a significant reduction of thrombus weight in thrombus weight in our vena cava model of venous thrombosis. And a similar thing we also saw in models of arterial thrombosis. A prominent consequence of loss of HSP47 in platelets was not only a reduction of thrombus size, but also a significant reduction in neutrophil extracellular traps. So loss of HSP47 translates into a reduction of neutrophil extracellular trap formation. So we wanted to clarify this in greater detail, and to do that, we exposed neutrophils to platelets isolated from bears at summertime or wintertime. And these platelets were either unstimulated or stimulated with collagen or thrombin, and PMA activation served as a control. And if you look at the control, actually, initially, you can see that in principle, neutrophils can, isolated from wintertime or summertime, can form nets to a similar extent. If you expose unactivated neutrophils to unstimulated platelets during summertime, there is a significant or almost significant trend towards increase of nets, also already without pre-activation of the platelets. But if you pre-activate summer platelets and expose them to summer neutrophils with collagen, you can actually see that there's a significantly higher tendency to form nets compared to the same scenario done during with samples isolated from bears during the wintertime. This accounts not only for collagen-activated platelets, but also for thrombin-activated platelets, indicating that activated platelets have a higher ability to trigger net formation in summertime, but not so much in hibernating bears during wintertime. So what is the mechanism behind? And it is known that T-shock proteins can actually activate target cells via toll-like receptor signaling. And it is known, on the other hand, that toll-like receptor 2 is one of the key targets of HSPs in general. So we tested this in several model systems. So we exposed neutrophils to HSP47 protein, isolated protein, in different concentrations. And you can actually see that this induces net formation. So this is in the absence of platelets. And of course, FMLP is a stronger activator of netosis in neutrophils, but a combination of FMLP and HSP47 dose-dependently further increases netosis. So in summary, HSP47 is, as a single protein, is able to support netosis in neutrophils. And how does that work? So to test the role of, as I said in the beginning, toll-like receptor signaling, We pre-incubated neutrophils with different toll-like receptor inhibitors, toll-like receptor 2 or 4 inhibitor, and also MyD88 inhibitor. And what we found in essence is that inhibition of toll-like receptor 2, but not toll-like receptor 4, abolishes HSP47 triggered netosis, not only netosis, this is not shown here, but CD11B expression. Similar thing was also seen for netosis. So HSP47 activates neutrophils via TLR2 signaling. So that is sort of the first part of my talk, and I would like to summarize that by looking at, which I think is a very intriguing model of hibernating bears, we came up with a new mechanism of how platelets can actually boost netosis, and this is via HSP47, which is expressed on platelet surfaces, which can activate TLR2 and neutrophils, which then leads to neutrophil netosis. And we think this is a very interesting cascade because it is used, or the downregulation of this cascade is used as a biological mechanism of prevention of thrombosis. And I should say that the mice lacking HSP47 in platelets have no bleeding phenotypes, making this a very interesting target. So I have now focused on platelet neutrophil collaboration in a sort of disease context, which we termed thromboinflammation. So I showed you an example of a patient with myocardial infarction. Also, I showed you the example of stroke patients having a very close correlation of neutrophils and immunity, leading to thromboinflammation, which boosts tissue damage through propagation of thrombosis. But of course, this collaboration is, as it is a conserved mechanism, probably not conserved because it leads to increase in cardiovascular events, but probably because it also has a positive aspect. And this collaboration, which is a physiological mechanism, is called immunothrombosis. And this immunothrombosis is known to be important for host defense against pathogens. And I don't want to go into much of detail, but just show you one example, which I think is very convincing example where this collaboration is relevant for host defense. And this is during infection. So we were interested in the ability of platelets to move. So when you expose, of course, platelets are known, considered to be cells that adhere when you have endothelial disruption, and then they are known to be activated and to form aggregates. This is sort of the standard view of a platelet. But when you look at platelets for a longer period of time and look at platelets that are exposed, for example, to fibrinogen here, which is known to coat, for example, inflamed endothelial cells, but also, of course, artificial surfaces like catheters that are implanted into the veins that are covered with fibrinogen. And you look at platelets that are exposed to these fibrinogen-covered surfaces over a longer period of time, then you can actually see that platelets are not just cells that activate and aggregate, but they can actually also accept or adopt a phenotype that is mostly known for inflammatory cells. So these cells can migrate, and they do so here on these fibrinogen surfaces by taking up the fibrinogen. So they collect the fibrinogen, and this fibrinogen here is shown in green. So it's a green tag fibrinogen. So during migration, the platelets sort of collect all the fibrinogen and concentrate the fibrinogen here on these areas which are called pseudonuclei. And this we found very interesting because fibrinogen is known to be sort of a biological glue also for any type of particles. One example, of course, are bacteria. Fibrinogen is known to trap bacteria. So we did an experiment where we added to dishes which are coated not only with fibrinogen, we also added bacteria. And this is shown here on the right-hand side. So these little sticks here are bacteria that are attached to a fibrinogen-coated surface. And in green and in yellow, we see platelets that are just as here migrating with this over this surface. And you can see actually that during migration, these platelets not only collect fibrinogen, but they actually also collect the bacteria that are attached to the fibrinogen. And actually they form these little bundles here. You can see this plate, for example, has like almost 40 to 50 bacteria bundled on the surface. And I don't want, as I said, to go into much detail because this has been published. We have analyzed this and we have found what the mechanism is behind this migration. It's mostly adhesion and then migration via alpha-2b beta-3 integrin. And then we have actomycin activation, which allows the forward migration of platelets. And this is essential for the collection of the bacteria, then bundling of this bacteria. And these bacteria then can handled over, so they are not internalized, they're actually exposed on the surface of platelets and then are handed over to, for example, neutrophils as professional phagocytes. So there's a very strong collaboration of these two cells, which leads to pre-cleaning of surfaces, inflamed surfaces covered with platelets, with bacteria, but also artificial surfaces during endocarditis could be an interesting target of this collaboration. And we have tested this in several models, in particular in the lung. We could show that the collaboration of these multiplatelets collecting bacteria, handing over to neutrophils is an essential mechanism for that protects from pathogen entry and pathogen spread into organs such as the lung, and is actually a highly protective mechanism, indicating that the collaboration of neutrophils and platelets is not only bad thing that drives thrombosis in the setting of myocardial infarction or stroke, but of course also has a positive aspect. And I just showed you one example of clearing of bacterial intruders. Since this collaboration has a sort of biological relevance, a positive biological relevance, it is not so surprising that the collaboration actually starts very early, so that there is an interaction of neutrophils and platelets not only at the effector function level, but also much early at the development phase. And this is the last part of my talk, where I would like to show you that neutrophils not only collaborate with adult platelets, but they actually are kicking in very early at the stage where platelets are developed and actually control the homeostasis of platelets and the development and the release of platelets from megakaryocytes. So platelets are generated in the bone marrow compartment, and the precursor of platelets are termed megakaryocytes. And this is something shown here. And actually this is a model that I learned in the lab of Uli von Andrian during my time at the Harvard University, which allowed us to image and to visualize megakaryocytes in the bone marrow compartment, a model established by Tobias Jund. At that time, these are labeled fluorescent-tagged megakaryocytes that are sitting in the bone marrow compartment next to vessels here, which are labeled in red. So this is a bone marrow vessel in the bone marrow sinusoid here, a megakaryocyte sitting in the extracellular space, but very close to the bone marrow vessels. And you see what happens. These megakaryocytes introduce very long processes into the intravascular space. And this actually was the first sort of evidence how platelets are really produced. It's another example here, megakaryocyte introducing two long processes here into the intravascular compartment here. This is sort of splitting up, and then at the tip of these long processes, platelets are being released. And another example is shown here, megakaryocyte showing releasing this very long process, and from the tip of this process, actually platelets are being released. And this in vivo imaging using 2-photon microscopy demonstrated that this pro-platelet model, which was proposed by Italiano and colleagues before, is actually the major mechanism of platelet production in vivo. So what we wanted to see is whether there is any role of neutrophils. Neutrophils are known to home to the bone marrow compartment and to be produced also in very close proximity to megakaryocytes. So it was an interesting question to address whether there is already a collaboration at this stage of development between these two cellular compartments. And if we use the model that I just showed to you, with two-fold imaging of the bone marrow compartment and staining megakaryocytes, in this case in red, and neutrophils in green, and the vessels in gray. In this case, you see again, this is a megakaryocyte sitting in the extracellular compartment here in black. So this is the compartment here, extracellular space. Here you see a megakaryocyte, and here you see 2 or 3 neutrophils binding to the area where the proplated process is extending into the intravascular compartment. So here is a bud, an initial bud, and then this elongates, and here the neutrophils interact very actively with this process, and then also at the tip there's an interaction. So this was the first sort of glance at a potential interaction that could play a role, of course, at this stage only descriptive. We tried to model this also in vitro, so we exposed megakaryocytes to neutrophils in an in vitro assay. And this is a megakaryocyte here with this proplated process, and you can see here a neutrophil that sort of binds to one of these processes, and then, as you can see here, these little particles leads to or appears to contribute to this function. Of course, again, just a descriptive observation, which is suggesting an interaction, but of course, at this stage, no causative evidence. But these two models triggered us to go in this direction a little bit deeper. And what we wanted to see is how depletion of neutrophils would impact on the processes of platelet development in vivo. So we went again to the in vivo imaging model that I just showed to you using two-photon microscopy of the bone marrow compartment of mice, and depleted intravascular neutrophils by an antibody against GM1, which does not go into the interstitial space in the bone marrow, but just depletes circulating neutrophils. And this was a very efficient model in terms of the depletion of neutrophils. And when we then in this model analyzed the appropriate length, so the length of these processes that are introduced from the megakaryocyte into the intravascular compartment, you can actually see that over time there is no increase in the length in mice that are were depleted from neutrophils, whereas in control mice, the length of the appropriate processes steadily increases over time, as I just showed you in the in vivo images. At the same time, this is sort of obvious, trivial, the proplated growth speed was significantly reduced in response to neutrophil depletion, and this translated into a prolongation of the time required for a proplated to release platelets. So it took longer to grow for the proplated processes, and it also took longer for the final release of platelets into the circulation when mice were depleted from neutrophils, from intravascular neutrophils. This translates into a reduction of circulating platelets. It's not a black and white picture, but you see a very reproducible and significant reduction in circulating blood platelets. And this is more evident if you look at the subset of reticulated platelets, which are considered to be young platelets, so platelets that were recently produced. If you look at, if you focus on this specific compartment of platelets, you see an even more pronounced reduction when you do neutrophil depletion, indicating or suggesting that neutrophils indeed support the release of platelets. We used another model as a control. We used MRP8 free deleter mice, in which we can delete all neutrophils, not only the intravascular neutrophils, but all neutrophils. And actually, the findings were very similar. So we found in these mice a platelet reduction of circulating blood platelets and a significant reduction also, again, in the reticulated platelets, supporting the concept that neutrophils indeed boost platelet release from bone marrow megakaryocytes. So what is the mechanism behind this? So to do that, we went back to the in vitro model system. We exposed neutrophils, megakaryocytes to neutrophils or known triggers of proplatelet production and quantified proplatelets in the supernatant of these megakaryocytes. So as a reference, because we use megakaryocytes in the absence of neutrophils, if you add sphingosine monophosphate, which is known to be a trigger of proplatelet release, there's a significant increase, and similar increase was also seen with neutrophils. So what we saw in the knockout mice, in the negative aspect with neutrophil depletion, we now see in the positive direction when we add neutrophils to cultivated megakaryocytes, an increase in proplatelet formation, further supporting that the concept of neutrophil-driven proplatelet release is actually valid. This depends on direct interaction, because if you separate megakaryocytes from neutrophils using a transwell, or just add the supernatant of neutrophils to megakaryocytes, there is no increase in proplatelet formation. Other cells like T cells, B cells, or monocytes are also not able to trigger proplatelet formation. So indicating that it's really a specific effect of neutrophils on megakaryocytes that leads to proplatelet formation, and that this mechanism involves physical contact. So what is the mechanism of this physical contact? The central mechanism appears to be CXCR4 expressed on neutrophils. So CXCR4 is the receptor for SDF1 alpha, and we know that particularly aged and activated neutrophils express high levels of CXCR4 on their surfaces. And if we used megakaryocytes and incubated them with neutrophils that are lacking this receptor on the surfaces, the increase in proplatelet formation seen with wild-type neutrophils was virtually abolished, substantiating the concept that indeed CXCR4 expression on neutrophils is critical for neutrophil-driven proplatelet formation by megakaryocytes. Similar thing was seen, or similar observation we made when we used not the knockout mice, but AMD 3100, which is an inhibitor of CXCR4. In contrast, ablation of P-selectin or integrin beta-2 or neutrophils did not have a similar effect. However, integrin ablation in neutrophils reduced the action, most likely because these neutrophils then were not able to bind to megakaryocytes any longer. So since CXCR4 appeared to be the most prominent signal required for neutrophils to trigger megakaryocytes to release proplatelets, of course, we wanted to see whether megakaryocytes express the ligand for CXCR4, which is called CXCR12 or SDF1 alpha. And this has been demonstrated before that megakaryocytes actually express CXCR12, and we confirmed this in this study, showing that megakaryocytes are very strong expressors of CXCR12. Here, all these large cells are megakaryocytes, and these small cells are other cells expressing CXCR4, but they are negative for CD41, so indicating that these are not megakaryocytes, but these cells are megakaryocyte lineage cells expressing CD41 and CXCR12. So megakaryocytes and their progeny are expressing the ligand of CXCR4. So we wanted to see whether when we knock out, we deplete neutrophils from CXCR4, would that translate in different phenotype in terms of platelet production in vivo? And to do that, we used MRP8 pre-deletion of CXCR4 in the neutrophil pool, and again used our in vivo model of photo microscopy of the bone marrow compartment and analyzed proplatelet formation. And as you see here, ablation of CXCR4 on neutrophils indeed, again, similar to neutrophil depletion, led to a reduction in proplatelet growth speed and a significant increase in proplatelet release time. And again, this translated into a reduction of the numbers of circulating platelets, in particular in a reduction in the number of reticulated platelets in the circulating blood of mice lacking CXCR4 on the circulating neutrophils. So how do neutrophils, once they encounter megakaryocytes, then trigger proplatelet formation? And we've performed a number of assays, and I want to focus on the ones that actually help identify the mechanisms, which show that in summary, local ROS signaling appears to be a major mechanism how neutrophils trigger proplatelets release from megakaryocytes. So again, we used the in vitro model of incubation of megakaryocytes with neutrophils, and we found that if we incubate megakaryocytes with neutrophil that are pre-incubated with an inhibitor of NADPH, which is the oxidase that leads to neutrophil ROS signaling, ROS release. This ablates the ability of neutrophils to trigger proplatelet release. Similarly, we found also inhibition of or addition of superoxide dismutase or knockout of NADPH oxidase on neutrophils leads to or ablates or abolishes the ability of neutrophils to trigger proplatelet formation. All with a different perspective, showing that release of platelets by neutrophils is triggered by binding first of CXCR4 to SDF1 alpha, and then activation of ROS signaling, leading to local release of ROS, which also again can be inhibited as shown here, but can also be mirrored. If you look at the megakaryocyte, phosphor-ERK signaling is increased if you incubate megakaryocytes with neutrophils, but not if you incubate megakaryocytes with neutrophils that are previously pre-incubated with apocynin, which inhibits NADP oxidase. The same is true if you look at phosphomyosin, light chain activate phosphorylation. So to summarize this part of my talk, I hope I could demonstrate you that neutrophils are an important mechanism that drives platelet release in the bone marrow compartment. They bind to the very early buds of megakaryocytes that are forming proplatelets. These neutrophil interactions can be visualized in vitro and in vivo. We showed in vivo that there is an interaction in vitro. We demonstrate there is a sort of a pulling and mechanosensing, as shown by phosphosine myelin phospho-MLC activation. This involves CXCR4 expression on neutrophils and expression of SDF1 alpha on megakaryocytes. And this interaction boosts the elongation and also the eventual release of proplatelets into the circulation. The outcome is an increased release of young platelets, and these young platelets are also called reticulated platelets. And this is interesting also from a sort of disease aspect, because increase of these young platelets is seen in patients and mice after a myocardial infarction. Here I show you the mouse example. So if you have a myocardial infarction, this leads to an increase in platelets and also in reticulated platelets. And we think that activation of neutrophils could be a mechanism behind this. Why is this interesting? Because it is known that the circulation on the rate or the frequency of young platelets in the circulation correlates with worse outcome with the increase of major cardiovascular events in patients with cardiovascular disease. And this is actually meta-analysis published very recently, summarizing data from several studies show that an increased frequency of young platelets in the circulation is associated with an significant increase, almost threefold increase in major adverse cardiovascular events after an index event of myocardial infarction, substantiating that abrupt release and sort of hyper release of young platelets can be something that has worse outcome. And that this is involving potentially the mechanism that I just showed you, so that the increased release of platelets after myocardial infarction could be indeed related to neutrophil activation. I want to show you in the last few slides. We again used our MRP8 Cree model with neutrophils depleted from CXCR4 and then expose these mice to myocardial infarction. And we saw that if you do myocardial infarction in these mice, the increase in platelet counts that I just showed you in wild-type mice is virtually abolished and reduced to mice that have no myocardial infarction. And this is specific, but particularly true for the compartment of reticulated platelets. So that indicating that when you have no CXCR4 expression on neutrophils, myocardial infarction triggered release of reticulated young platelet is virtually abolished. And this translates into a reduction of secondary events. So what we did here, we exposed mice to a first myocardial infarction and looked at mice having CXCR4 expression or having no CXCR4 expression on neutrophils and compared thrombocytes when we did a secondary myocardial infarction. And in wild-type mice or pre-control mice, there was an increase, of course, in thrombus formation after 10 minutes of, this is a model where we did carotid injury. And this increase can be virtually, also at least significantly reduced if you have mice that have no expression of CXCR4 on neutrophils, and this thrombocytes overall significantly reduced, substantiating the model that activation of neutrophils during myocardial infarction triggers increase of young platelets that then can boost secondary ischemic event, thrombotic event, because these platelets have been shown to be more reactive. And so ablation of CXCR4 or targeting CXCR4 after myocardial infarction could be a promising strategy to prevent future cardiovascular events. And with that, I would like to summarize that last part of my story, that neutrophils can activate proprietary formation, so they are sort of fine-tuning proprietary release, the mechanism involving CXCR4 and SDF1 alpha interaction. And this leads to a release of hyper-responsive platelets that can activate thrombotic signaling in patients with cardiovascular disease, supporting worse outcomes. With that, I'm at the end of my talk and would like to thank, of course, all the lab members, in particular, Tobias Petzold, Leo Nicolai, Helen, Shikaba Ankerholt, Konstantin Stark, Manuela Thielen, Florian Gärtner, Rainer Kaiser, Ina Saleh, and Kami, and Wen Fu, who contributed data to the talk I have given today. Of course, I thank all the other lab members. And in particular, thanks goes to all of our collaborators. I mentioned the Scandinavian Brown Bear project, which was essential for the hibernation study, but also, of course, all our other collaborators and the funders that supported our work in the last 15 years. Thank you very much.
Thank you, Dr. Massberg. That was a fantastic presentation, very interesting. And we're now opening up for Q&A. And for those of you who are interested, please submit questions to the Q&A box. Please be mindful that though this talk was very focused on platelets and neutrophils, which I, of course, really enjoy, Dr. Massberg is also a cardiologist and would probably be able to answer some of your questions more on a clinical research side as well. But maybe I can start with a few questions that came up. And as you know, thinking about your last comment here, your last discussion point on the plucking, there are other mechanisms too that have been described where neutrophils interact with megakaryocytes with embryopoiesis, where the neutrophils seem to swim through the megakaryocytes and then go out. Do you think that process has anything to do with what you have described here with the plucking, or is that a completely separate kind of mechanism?
It is a separate mechanism. Actually, in the model that I showed you, we can actually also see the transmigration of neutrophils. But numerically, this is a very rare event. So if you look at area of interest, you will eventually see one neutrophil migrating through a megakaryocyte, whereas you have hundreds of neutrophils interacting with the appropriate processes. But it's an interesting question. And of course, we looked at a sort of baseline or physiological scenario. It could be a different picture if you look in a disease setting. So then this process of transmigration could be increased eventually. And it's actually a point.
Right, and do you think there's any difference between where the megakaryocytes are located, that is in the bone marrow or in the lung, or do you think location matters for these mechanisms?
So we focused on the lung, on the bone marrow, and we did the imaging. But of course, the outcome of platelets in the circulation can derive from both sources. So in essence, we cannot really discriminate between lung and bone marrow sources. But there's recent evidence supporting the concept that most of the sort of steady state production of platelets occurs in the bone marrow compartment. So I would argue that based on that observation, the major way of action is actually in the bone marrow compartment. However, I cannot exclude that similar mechanisms also work in the lungs.
Exactly. I'll just do one more before I open up for the team here. Thinking about neutrophils, nets, platelets forming these thrombus, why in your mind is not these nets degraded quickly? Because we should have DNases in circulation. What is making them so stable that they remain or retain sufficiently long, at least, to cause a thrombus?
Actually, I don't know, but it could be that the clot formation sort of provides some sort of protection also against degradation of the nets. So the longevity of nets is different if you compare nets in the absence of coagulation compared to presence of coagulation. So it could be that coagulation avoids some of the degradation. But one aspect I think is that probably it's not necessary that the nets live forever, but there's continuous recruitment, ongoing recruitment of new neutrophils. So probably also there's steady state degradation and new production of neutrophils of nets in the process of thrombosis.
And do you think that the presence of, let's say, anti-DNA antibodies or anything like that then prevents degradation of nets could be an amplifying factor in diseases such as lupus?
Yes. I would imagine so, yeah.
Excellent, thank you. And you said you wanted to review the questions, or would you like for me to share them with you?
I open it. So
Excellent.
Keith Alkin asked whether the effect of HSP47 on net formation looks, or he mentioned that it looks modest compared to other stimuli. And he asked whether HSP concentrations are increased in patients with systemic autoimmune diseases and net formation and clotting. So the effect, I agree, it looks modest if you look at sort of isolated HSP47. But the effect, if you look at the effect of HSP47 deficiency on net formation, it's not so modest. So of course, if you add something recombinant to cells, it is a completely different scenario compared to when you have HSP47 on a surface, such as the platelet surface. So I would say it's not a moderate, it's a strong inducer of netosis, but of course, it's not the only. Inflammatory signals such as FMP1 is a stronger inducer of netosis. So is the net formation in the setting of bacterial infection. Of course, the level of net formation is completely different from the level of net formation in thrombosis. So it is compatible with the major role in the net formation in the setting of thrombosis. But of course, when you have sepsis or other very strong inflammatory signals, this may override and have a much stronger impact on netosis. Then, can neutrophils present platelet-derived antigens? Yes, they can. There's some evidence that actually membrane particles can be expressed, but also other particles released by platelets. This is an interesting topic which we are not really focusing on, but there is some literature on that. And it could be that this is also a mechanism how neutrophils become activated, and actually activation is also propagated to other subsets of inflammatory cells. Jose Lopez, hello. Thanks for listening. Do patients with selective neutropenia have statistically lower platelet counts? It's a very good question. It's very difficult to answer because there are patients that have sort of a congenital neutropenia, and these patients have a lower counts of platelets. In particular, if you look at the newborn platelets, there's a reduction. Many other scenarios where you have neutropenia, like in certain stages of sepsis, for example, also have thrombocytopenia. But there, of course, it's very difficult to discriminate between increased consumption as opposed to reduced production. But in the congenital scenarios, there is an association between the number of neutrophils and the platelet counts. And then we have another question: does the inflammasome play a central role in triggering the inflammatory process with DAMs from damaged endothelium triggering activation? Yes, it does. DAMs released from, for example, from platelets, but also, of course, from endothelial cells lead to neutrophil activation. And this has been demonstrated to be one of the major mechanisms of also neutrophil activation and net formation. Also in the context of thrombus formation. Any change of plasma von Willebrand factor level in bears in summer versus winter time? We looked at that, but we did not find a significant difference. Also not in the, when we looked at the multipolarization of von Willebrand factor.
Wonderful. If you don't mind, I have another follow-up question on your early discussion, linking it to some of your last findings. So you said in this model of myocardial infarction where you had the CXCR4 deficiency, and you saw a reduction in thrombus size. Were you also able to look at the cellular composition of that thrombi and seeing whether reduction of neutrophils also affected certain other immune cells? Kind of asking the question, are neutrophils important for recruitment of immune cells?
I would expect yes. We did not look at that. We looked actually at later stages, and there we found that monocytes are important to recruit subsets of neutrophils that are essential for thrombus degradation. Whether neutrophils are really essential for recruitment of like monocytes in the earlier stage of thrombus formation, we don't know. But I could envisage several mechanisms that could do so. Yes. Yeah.
And to that point, do you expect to see different cellular compositions, whether it's a thrombi formed on the arterial side versus the venous side?
Yes. So this is something we look at in patients with a stroke. Stroke patients, of course, there is two sort of different cohorts: patients that have atherosclerosis of arteries that develop thrombi, and patients that have atrial fibrillation, which more or less resemble the venous thrombosis. There we see that the contribution of platelets is different. So in the arterial setting, we have more platelets and less coagulation. We do have neutrophils, but the neutrophil numbers are a little bit lower, which is probably explained by the dynamics of thrombus formation in the arterial setting. In the venous setting, we have much more neutrophils. It's a more inflammatory phenotype and also more nets, presumably because these thrombi develop over longer periods of time. And we also have more fibrin in these thrombi. And this also appears to be a determinator of age, or vice versa. So if you have a lot of fibrin, a lot of neutrophils, these are typically the older thrombi in the venous setting.
Excellent. Well, thank you so much. And if no further question, thank you so much, Dr. Massberg, for a wonderful presentation and for taking your time this late Tuesday evening from home or from Germany. Again, thank you so much and have a wonderful rest of your day.
For having me, and good afternoon or good morning to you. Yeah.
Thank you.